cutest little champions, I welcome you all to score 99% in 99 days. Today is day 27, today is Saturday, and Saturdays are for revision. And who am I? I am Jasmine Joy, I deal with biology, and today we are going to revise a chapter that is heredity. And I want to tell you one very important thing. If you are someone who is looking to understand the concepts, please, I would request you go watch the videos which was given down on Monday and Tuesday. It will clear down your concepts. We thought of having a revision on this particular date because sometimes you don't find time for revision. So we thought, why not help you with that? So this is solely meant for that purpose. We would be going really fast. So you can imagine me as your best friend, okay, who help you with the revision in the last minute before just the point of exam. So I am that person for you. Now Soumik sir will also be dealing with physics, okay, he would be revising physics, human eye and the colourful world. It will be after I am done with the revision. Works? So let's get started, children. Over here, the first thing, what do you mean by heredity? Heredity is the transmission of what? Is a process of transmission of characters from the parents to the offspring. This whole process where characters are being transferred from the parents to the offspring, this is known as heredity. Now going ahead, when you say you are transmitting characters, what are these characters known as? In scientific term, you call this as traits. Okay, you call them as traits. Now traits can be of two different types. One are inherited traits, another one are acquired trait. As you can see over here, if you see at this picture really carefully, you will be able to find that eye color, ears, shape, height and everything. These are the examples of inherited traits, something that you can inherit from your parents. Now for a second, you just imagine Jasmine went to gym. Now I went to gym, I have muscles now. Can I expect that the progeny that I will be having will also be having muscles? No. Because this is an example of acquired trait. For example, I colored my hair. Can I expect my progeny will be having multicolored hair? No. Again, if I have a tattoo, can I expect my progeny will be having tattoo? No. So these are examples of acquired trait, something that you are acquiring in your lifetime, and these traits cannot be inherited. So inherited are the ones that comes over here, acquired are the ones that comes over here. Now if you want to write the definition, you can look at this particular slide. Going ahead, whenever we say that basically we have inheriting characters from our parents, why do you think we look like our parents? What is the reason for that? Because they have undergone something that is known as sexual reproduction, in which two parents are involved. Okay, the mother gives the ovum, the father gives the sperm, together they come fuse, and now we have the zygote. So over here, I am inheriting both of their characters. Over here, in the case of sexual reproduction, what is happening, children? I am inheriting both of their characters. From the mother, from the father, I am getting the DNA, which is why I am not looking exactly similar like my parent 1 or probably parent 2. I am a mix of both of them. So here basically, don't you think there is a variation happening? There is variation happening. I am a mix of both of my parents. I am not exactly similar. If I was exactly similar, I would say there is no variation. But I would say in sexual reproduction, there is variation because two different parents are involved, two different genetic material is being involved over here. There is one more reason, one more very big reason for variations to happen in sexual reproduction. And what is that reason? Basically, when I say that I am getting their DNA, it's not like they are giving their own DNA to me. They are making a copy of their DNA. The cells which are involved, they are making a copy of their DNA, and that copy is given to me. So when this particular DNA is being copied, so for at that particular point of time, now what happens? This DNA will open up. It will create a new copy over here. This strand will have a new copy over here. This strand will have a new copy over here. And now, one will be kept with the parent, one will be given to me. So during this process, what do you call this process as? You could not call it DNA copying. You call this as DNA replication. You call this as DNA replication, my dear children. And during DNA replication, there are so many errors that can happen. During DNA replication, there are so many errors that can happen. So mostly our body senses it. There are enzymes that take care of it and they kind of repair it. But sometimes these errors sustain. And whenever there is a change in the DNA sequence, which is over here, you say that it will arise, it would lead to variations. It would lead to variations. Now these variations, these changes can be good and can be bad. These variations can cause diseases. These variations can help you survive as well. So there are two reasons why in sexual reproduction you will be having more variations, okay, or you can give rise to more variations. First thing, the DNA copying errors will be there. Second thing, the major thing, that is here two parents are involved. So two parents involved means two genetic material coming together, which gives rise to variation. If only my mother was contributing the DNA, my father was not, I would have exactly looked like my mother. But that's not the case. Two parents are involved, there is fertilization happening, which is why there is more amount of variation. This is a very important question, why sexual reproduction gives you more variation. So because of the contribution of genetic material from two different individuals, as well as due to replication errors. Now I want to ask you a question. If sexual reproduction has more amount of variation, what about asexual reproduction? Does it have variation? Does it lead to variation or not? In asexual reproduction, if you remember my children, there is only one parent involved over here. There is no fertilization of gametes. There is no fusion of gametes. There is only one parent involved. For example, an amoeba. An amoeba divides into two daughter amoebae. An amoeba is dividing. An amoeba. There is only one amoeba over here. So this particular amoeba will be contributing its own DNA in there. There is no mixing of genetic material over here. But I can still say variation will be still there. Why? Because obviously this amoeba, when it gives us DNA, it has to copy again. There will be certain copying errors, there will be replication errors, which is why I would say there will be a little amount of variation. There are chances that little amount of variation will still be there. But it won't be as big as sexual reproduction. Over here, two parents are involved. Over here, just one parent only being involved. Works? Cool? Now, one very important thing which is given in your NCERT, that is an example of a bacteria. So how do you think variation can help in evolution? How do you think variation can help in evolution? Evolution as in creation of new species. Okay, or probably how do you think variation help in continuity of the species? So basically just think of two bacteria. There is bacteria 1 and there is bacteria 2. Suppose this particular bacteria had some variation. It's some changes in the DNA which lead to some variation in them. This particular bacteria does not have that particular variation. Now, a heat wave or something came like that. Okay, but since this particular bacteria had this variation, it was able to survive. It was able to survive, whereas this was not able to survive because this didn't have the variation. Now, you tell me, only this guy is responsible for what? Continuity of this species? Continuity of the species. If this variation was not there, will it be able to continue its species? No. So this is why variation is really important. Now these variations, obviously there will be more variations accumulating which would lead to evolution. Now slowly what will happen, this guy would be very different from this particular guy. It will have so many variations coming along its life. Okay, so that's how evolution happens. The nature selects the one that is perfect. Over here, this heat wave, this is a nature selection method. This heat wave is nature's thing, right? We didn't create it. And now the one who was surviving will survive, it will continue. One that was fit, it will survive, survival of the fittest. You get it? I hope it's clear. So always remember, sexual reproduction gives you more amount of variations. Now, talking about Mendel. Okay, talking about Mendel or the genetics part. So listen to me very carefully. Mendel, Gregor Johann Mendel is his name. Okay, what is his name? Gregor Johann Mendel is his name. He is known as the father of genetics. He is known as father of genetics. He was the one who was studying what is happening between parents and offspring. Are the parents giving some features to the offspring? How do they look similar? So without doing the experiments on animals and stuff like that, he actually chose a plant which is known as garden pea. What did he choose? He chose garden pea. It is also known as Pisum sativa. It is also known as Pisum sativa. This is a scientific name. This is the actual normal name. Okay. And according to him, he gave out 3 very important findings. When he did experiment on garden pea plant, okay, he gave out certain very important findings. But before that, I want to tell you one very important thing. Where is it? Why did he choose garden pea plant? Because garden pea plant, no, it was able to reproduce really fast. Okay, it was an annual plant. Annual plant as in it will stay for an year. So basically it will reproduce in a year, and then, so you can see the progeny. For example, I want to check if the parents are giving the features to the progeny. I can clearly see it within 1 year itself. I don't have to wait for years to see that. And it was very easy to grow them. It was very easy to grow them. Apart from that, they had 7 contrasting traits. What do you mean by 7 contrasting traits? Basically, for example, if I'm talking about height, in them there are 2 different types of plants. One were tall, the other one were dwarf. This is the exact opposite of things, right? Tall and dwarf. I can clearly distinguish between them. Similarly, they had 7 such characters. One was height, other one was seed shape, seed color, pod color, stuff like that. How many? 7 contrasting traits were there. So which is why he chose the garden pea plants. Okay. Now, according to Mendel, what did he come up with in the findings? He said that all the traits that you see, for example, height, it is specified by some heritable elements which are known as genes. He didn't say genes, he called them factors. What did he say? He called them factors. He said whatever, for example, you're talking about height, this particular height is being coded by something. He didn't know genes back then, and he said it's there in our genetic material. This particular thing is there in our genetic material. Okay, so it's not like randomly you get it. There is something in our genetic material that will be coding for a particular trait. Now, he also said one more thing. Genes come in different versions. He said factors comes in different versions, or what do you call them? Alleles. What do you mean by that? Basically, when I say there is height, when I say there is height, children, height is of 2 types. There is tall, there is dwarf. So, there is a gene for height. There is a gene for height. Now, for gene for tall one will be different and dwarf one will be different. Gene for tall one will be different, for dwarf one will be different. For example, I say capital T gene is for the tall one. Small t gene is for the dwarf one. So, basically, can I say these are the 2 different types of the genes only? There are 2 different types of this particular gene. What do I call these different types as? I will call them alleles. I would be calling it alleles. So, different versions of genes are known as alleles, my dear children. Now, alleles are basically of 2 types. One a dominant allele and the other one is what? Recessive allele. So, for example, we all have a pair of chromosome. We have 23 pairs of chromosome. Okay, we have 23 pairs of chromosome. Similarly, plants also have pairs of chromosome. So, this is a pair. Okay, over here I have a gene. Over here I have genes for what? Height. Now, they are of 2 types. One is capital T, other one is small t. Now, in this pair, if someone is having this pair and this particular plant is tall, that means only one allele is able to express itself. The other allele is not expressing itself. The other allele was for dwarf. It was not expressing itself. That means, can I say that this allele that you see over here is dominant over the small allele? Yes, I can say. So, this allele I will call it a dominant allele. The one that does not show itself, does not show its property, it is known as recessive allele. Okay, it will show its property only when the condition is like this. For example, I have a chromosome where both of them are small t. There is no dominant allele to overshadow its effect. At that particular time, it will show its effect. Okay, what do I call this condition as? And what do I call this condition as? Whenever I say there are 2 alleles of the same kind over here, I will call this as homozygous condition. I will call this as homozygous condition. For example, over here you have this. Similarly, there is capital T, there is capital T. Still, the alleles are same. Still, I will call it homozygous condition. This particular plant who have a pair of allele like this, it will be dwarf because there is no dominant allele. Over here, when I see capital T, this is dominant. So, anyways, it will be tall only. When I say this condition where both the alleles are different, I will call it heterozygous condition. Heterozygous, but it is heterozygous tall or dwarf? It is tall because there is one dominant allele over here. So, I will call this heterozygous tall. So, this is heterozygous tall, this is homozygous tall, this is homozygous dwarf. Works. So, the alleles are of 2 types, dominant alleles and recessive allele. Dominant allele expressed over the recessive alleles. Recessive alleles are only expressed when there is no dominant allele present. Got it? So, each of our traits or each of our characters are being specified by some factors, by some things that are known as factors, which is there in our genetic material, and these factors come in different forms. Okay, one will be dominant form, other one will be recessive form. Now, what do you mean by dominance and recessive? How we can say that this is dominant or this is recessive? Basically, these things that you see, this is our DNA. Okay, which is condensed and formed, it forms what? The chromosome. I will say there is a gene over here, and this gene is coding for some protein, and this protein is kind of very big, it's able to show its effect. And I am saying this particular chromosome has DNA with a gene coding for the same protein, but the amount of protein is kind of less. So obviously if protein is produced more, its effect will be more, right? This protein is obviously produced less, so effect will be less. So this is why I'll say this is recessive and this is dominant, which is why, okay, it is able to show its effect. I hope this works. So these were the things said by Mendel. Now going ahead, one more thing that you have to remember, this pair of alleles or the genetic representation is known as genotype. So whenever I say that this is a condition, what is a genotype? This pair of alleles you have to remember, it will be capital T, capital D. This is the genotype, this is a genetic makeup of an organism. Now the things that I can see from outside, the genetic makeup I cannot see. I cannot see the genetic makeup which is there in my body, okay, it's just there. I can see the character that I am having. For example, am I tall or dwarf? That is something that is visible. For example, a plant that has this will be obviously, it is homozygous tall. Okay, there's dominant alleles only, so it will be tall. So this feature that you can see, physical feature that you can see, physical feature, phenotype, physical, can remember like that. This is known as phenotype. So if you're asked, you're given this and you're asked, what is the phenotype of this particular plant? It will be tall. If the condition was like this, and you are asked, what is a genotype? What is a genotype? Genotype means genetic makeup. The genotype is small t, small t. What is the phenotype? How does it appear to be? It will be dwarf children. So this is the phenotype. Get it? Now, tell me, okay, just one more thing. Tell me what will be the genotype of this. What is the genotype? Genotype is genetic makeup, that will be capital T, small t. What is the phenotype? This is dominant over this, so it will be tall. Got it? Works? Now, Mendel, he came up with all this thing when he was trying to cross the plants. He was trying to cross the plants. So whenever he was studying only one particular trait, for example, he was studying just about height, and he was seeing what all things he could find with just the height. This type of cross is known as monohybrid cross. So it involves the study of inheritance of one pair of contrasting characters by crossing between two parents. So he was just studying about one pair of contrasting characters. What do you mean by one pair of contrasting characters? For example, tall and dwarf. He was studying about that, but just one pair. He was not studying height as well as the color of the seed. No. He was just talking about one particular trait at one particular time. This is known as monohybrid cross. Now there is something that is known as dihybrid cross, where he was studying two characters at the same time. He was talking about the height and also the shape of the seed. This is known as dihybrid cross. The study of inheritance of two pairs of contrasting characters is known as dihybrid cross. Works? So first we will go with the monohybrid cross and then we will be going ahead. So one more very important thing, how traits are expressed, or I think I'll talk about it later. I'll talk about it later. Okay, just remind me. Cannot remind me? Okay. What should we do? We will do it now only. So how traits are expressed? No, I should be doing later. You will get confused. So I told you he experimented on pea plants, right? Why did he experiment on pea plants? As I said, it was simple to grow and keep up with. There was large number of characters which are distinct and contrasting. Okay, I told you 7 pairs of contrasting characters are there. Then because the pea plant is an annual plant, it is possible to study many generations of the plant in a shorter period. So I don't have to wait for years to see the results. Okay, peas are naturally self-pollinating, but they can also be pollinated by other plants. So I do not need to pollinate the plant. I do not have to put the pollen grains over there. They are self-pollinating. They will pollinate among themselves only. So they have a flower which are known as bisexual flowers. So they will be having female and the male reproductive system on the same plant, which is why it will be self-pollinating. So it is kind of easy for me to grow a plant like that. So these are the 7 pairs of contrasting characters. Apart from this, I think we do not have to study this, so you can delete this particular thing. Instead, you can write the character as flower color. In flower color, why is it not working? In flower color, violet is a dominant trait and white is the recessive trait. This is what? Flower color. So these are the 7 pair of contrasting characters. Height, in height, tall is dominant and dwarf is recessive. In seed shape, round is dominant and wrinkled is recessive. Obviously, you will prefer round seeds to be eaten, right? So this is dominant. In seed color, even though we eat green color pea, okay, but actually yellow is the dominant color and green is the recessive one. And now, the flower color, violet is dominant and white is recessive. In pod shape, inflated, as in it is full. Okay, it's not constricted, it is the dominant trait. Constricted one, the dry one, is the recessive trait. Okay, pod color as in the chilka's color, the outer covering which you take, it is usually green in color, green is dominant and the yellow one is recessive. Now flower position, there are two type of flower position, one can be like this, one can be like this, where flowers are coming from the sides. This is known as axial. This is dominant. And the flowers, if a terminal, it is known as recessive trait. So please understand, these are the dominant traits, these are the recessive traits. How many of them do you have? 7 of them you have. So whenever I say tall is dominant, I will use capital T for denoting it. I will use capital T for denoting it. I will use small t for denoting the recessive one. I won't say S. I would use the small version of the same letter. Okay, so here we have round and wrinkled. So I will use capital R for this. For wrinkled, I will use small r. Instead of using W, I will use small r. For yellow color, I would use Y. Over here, green color, I would use small y. Because yellow is dominant, which is why I used Y, capital Y over here and small y over here. For violet, I will be using capital V. Over here, for white, I will be using small v. Got it? So this is how things work. Please use it accordingly. There's only one difference, I think. Okay, I think in the case of this thing, round and wrinkled, sometimes small w is also being used, so do not be worried. Okay, accordingly you have to understand, but whenever you say capital letters, just understand it is a dominant trait. Fine, works. Chalo. Now let's see what did he do with the monohybrid cross. So there are different type of scenarios. I told you, basically you get your chromosomes from your parents. You get the chromosomes from the parents' children, okay, or the genetic material from your parents. So I told, for every character you have a pair. For every character you will be having a pair of chromosomes. So when gametes are being formed, for example, ovum is being formed in the case of human body, in the human female body, what happens is that this pair will segregate and only one can go into the gamete. Into one gamete at a time, only one can go. So from this, either this can go or this can go. Suppose this has went. So I will have capital T in the gamete. Now what happens? Suppose I got it from the mother. Okay, this is how it looks. So I have capital T, one chromosome from the mother. From the father also, father will also be having a pair like this. From the father also, I will be getting only one, which can be any one. Suppose I got small t. Suppose I got small t. But anyways, I want to make you understand, that is, in the gametes, half the number of chromosomes will be coming. So in the parents, if they have 46 number of chromosomes, in the gametes there will be only 23 chromosomes. From every pair, from every 23 pair, you will be getting just half of it. Okay, from a pair you will be getting only one chromosome. So can I say, from 23 pairs of chromosome, I will be getting half of it. Will that work? Okay, just a second guys, something went to my eyes. Okay, I think I will be fine. Yes. Just give me a second. Just give me a teeny second. Okay, I'm fine now. So basically, one chromosome from the egg that comes from the female. Okay, there will be one chromosome as you saw there. Similarly, one chromosome coming from the male. Over here I have capital T, over here I have small t. Okay, so what will be the combination that will be forming a new pair now? So in the gametes, so in the parent we had 46. In the gametes we have 23. Okay, 23 from papa, 23 from mama. Both comes together, again I have 46, and the normal state is fine. So only the gametes will be having half the number of chromosomes. You have to understand that. Okay, now let me explain. So I got a chromosome from here, a chromosome from here, again I have a pair. Okay, now over here what will be the character, children? This will be tall. Why? Because this is capital T. Capital T will be dominant. So it will be capital T small t. What is the genotype? Capital T small t. What is the phenotype? It is tall. Similarly, next condition, I get capital T from both the parents. Over here, what will be the genotype? Capital T, capital T. What is the phenotype? It will be, appearance will be tall only. Okay, this is heterozygous. They are different. This is homozygous, tall. And homozygous, I also call it pure breed. Whenever it comes to be homozygous, I also call it pure breed. Okay, now next thing, over here I get small t, small t. What is this? Genotype. Genotype will be small t, small t. What is the phenotype? There is no dominant allele, so it will be dwarf. What kind of dwarf, children? It will be homozygous. It is same. So dwarfness will be only shown in homozygous condition. Recessive characters are only shown in homozygous condition. Okay, please remember that. This is also pure breed dwarf. Works. So these are the different scenarios that we have. Now we will see what actually Mendel did. So Mendel took a pure breeding tall plant. Okay. So there is no mother and father in plants. So he took pure breeding tall plants and pure breeding dwarf plants. Now the gametes can be either this, either this. Anyways, whatever you do, it will be capital T, capital T. Here also, whatever you do, it will be small t only in the gametes that are being formed. Suppose this gamete fused with this gamete, this fused with this, you get capital T small t. This fused with this, you still get capital T small t. Whatever you do, you will get capital T small t. So for the 4 progenies that could be produced, all of them will be having capital T small t. So what is the genotype in F1 generation? This is the first generation. What do you call this? F1 generation. And this is the first generation when you breed pure breed of tall and dwarf plants, or homozygous tall and dwarf plants. You see the genotype is capital T small t. What is the phenotype? How do they appear? Capital T is there, so it will be dominant over this, so hence it will be tall, my dear children. Now Mendel was like, how can I be sure whether it is tall like this or it is tall like this? Because anyways I cannot see, I can see just the physical character. Right, so I have to come to a conclusion. How can I do that? So basically what he tried to do is, he did something that is known as selfing. So in between the progenies, he tried to breed them. So if you are crossing a plant that is tall like this, what do you get? This is the parent, this is the gamete, and this is the F1 generation, suppose. So F2 generation. So over here you have the gametes as capital T, capital T. Here also gametes will be capital T, capital T. Whatever you do, you get capital T only, right? Now when you fuse, all of them will be this like this only. Get it? So all of them in F2 generation will be tall. In the next generation, all of them will be tall. This would be the conclusion. But if it is like this, the scenario is going to be different. Let's see the next scenario. So basically, he took the F1 progeny, he did selfing to find out whether it is homozygous tall or it is heterozygous tall. So it looked like this. Ayyo, where is it? So capital T small t, capital T small t. Over here the gametes are of 2 types. Either it can be capital T or it can be small t. Here also it can be capital T or small t. If this and this fuses, you get capital T, capital T. Okay, if this and this fuses, again you get something else, capital T small t. Here you get capital T, capital T. Sorry, I messed it up. So here you get capital T, capital T. Here you got capital T small t, and here again you got, when you fuse this with this, you get capital T small t again. When you fuse this with this, you get small t small t. So I see different combinations. Over here I was able to find only one. All of them were tall. Over here, when I see, not all of them will be tall. You see, this will be tall because it is capital T, capital T. This will also be tall because this is dominant. But this will be dwarf, my dear children. That means that the thing that were being born, what this? It was not this. Okay, so 3 of them will be tall, 1 of them will be dwarf. This is the phenotype, physical appearance. 3 of them will be tall, 1 of them will be dwarf. So what is the phenotypic ratio? What will be the ratio, children? It will be 3:1. So how will you write it? You have to write like this: T tall and 1 dwarf. Like that you have to write. This is the phenotypic ratio. Now if you are asked the genotypic ratio, what is the genotype? So you see there is only one that has capital T, so I'll write 1. 2 of them have capital T, small t, so I'll write 2. Over here only one of them has small t, so this is again 1. So what will be the ratio? 1:2:1. That is the next slide. So where you see this will be 1, this will be 2, and this will be 1. So how do you write it again? 1, 2, and then 1. You have to mention it properly, otherwise you are going to lose your marks. So please do not do that mistake. So according to F2 progeny, this is what he got. Please remember the phenotypic and the genotypic ratio of F2 generation. In F1 generation, there is nothing to remember like that. Okay, works? Will you be able to remember? This is what is known as mono-hybrid cross. Okay, and what did he understand after doing the mono-hybrid cross? So these were 2 laws that he gave after doing mono-hybrid cross. Let's see that. So if there are two alleles coding for the same traits, and if one is dominant over the other allele, then it will show up in the organism while the other won't. That is, if there are two alleles, suppose, okay, coding for the same trait. Yes, they are coding for the same trait, that is height. If one is dominant over the other allele, then it will show up in the organism. So it was dominant, which is why it was visible in F1 generation. All of them were tall. Okay. And the other one will not, that is small one will not. This is known as law of dominance. This he got from doing the monohybrid cross. The second thing that he got was, when an organism makes gamete, each gamete receives just one gene copy or one allele. So for example, this is an organism. Now if I want to make gamete out of this, I will, in a gamete, there will be only one chromosome coming. It can be either capital T or it can be either small t. So over here, I see that the chromosomes are basically segregating, or I'll say the alleles which are there on the chromosomes, they are segregating. So when an organism makes gamete, each gamete receives just one gene copy or one allele, which is selected randomly, and this is a random process. Anything can be selected. Either this gamete can have a capital T or it can have small t. It is a very random process. And what do you call it? This is known as law of segregation. Okay, this is known as law of segregation. So these two laws were put down by Mendel after doing the monohybrid cross. When he did the monohybrid cross, that's when he understood things like this works, because he saw it's a very random process. I was not able to see any tall plant in F1 generation, but I saw them in F2 generation. That means the recessive allele was still there. The recessive allele was still there. It was able to show itself again because of law of segregation that was happening. So it was segregating properly. Okay, it was a random process that was happening. Similarly, when I got two such things, I got the small plants, dwarf plants in F2 generation. Works. Then comes something else which is known as dihybrid cross. Okay, then comes something else which is known as dihybrid cross, my dear children. What happens in dihybrid cross? I am going to talk about two different traits at once. So basically, suppose there is round. Okay, round is mentioned by this, and there is yellow, which is mentioned by white. Sorry, Y. Similarly, opposite of round is wrinkled. So I will use smaller, and then I am using green. Opposite of yellow is green, so basically I get small y. So as you can see, round, yellow, there is a plant which has round seeds and yellow seeds. There is a plant that has wrinkled seeds and green ones. So I am crossing a plant that has round yellow with wrinkled green. So this is the dominant trait, these are the recessive ones. So how can I write it? I have to talk about two traits together now. So capital R, capital Y. This will be the normal one. Over here, small r, small y. Dihybrid cross. I am going to talk about two characters at once. Okay, now how is the chromosome? How can you imagine the chromosome? So just suppose this is a pair of chromosome. One has capital R, the other one also has capital R according to this. Similarly, one has capital Y, another one also has capital Y. Over here, one has small r, other has also small r. One has small y, other one also has small y. Now I have to form, this is the parent, this is the parent. I have to form the gametes. So what will be the gametes, children? From this pair only one guy can come in. From this pair only one guy. Either of them, whoever comes, it will be capital R only. From this pair also one guy can come in. Either capital Y or capital Y. Whatever I do, it will be capital Y. So can I say this is the combination that I get? Okay, suppose if you are not able to understand, one chromosome from here and one chromosome from here, it is coming. Okay. Now over here, similarly, small r can come or small y can come. So it will be either small r or small y. Okay, now I have to fuse them. Gametes have to be fused. Whatever, there are so many gametes can be formed, but all of them will be having this only. Now when I fuse the gametes, that is in F1 generation, what will I find? In F1 generation, what will I find, children? This and this gamete, if it comes together, can I say that this and this chromosome are coming together and these are coming together? So it will look something like this. Capital R, small r. Okay. Capital Y, small y. So can I write capital R, capital Y, small y? This is the F1 generation. Okay, how do they look? This capital R is dominant over this small r, capital Y is dominant over this small y, right? So this will be round and this will be yellow. So in F1 generation, all of them, they show the dominant character. They show the dominant parent's character, that is round and yellow. But still, now Mendel was confused whether it is homozygous like this or it is heterozygous like this. He can only see it round and yellow. So to confirm that, he had to make a cross again of this particular, he had to cross the progenies which were like this. Okay, and then what will he get? Let's see that. So basically, how will I make it? I'll make it here. So I have capital R. Got it? Over here, now the chromosomes are like this. Capital R, small r, capital Y, small y. Similarly, capital R, small r, capital Y, small y. Now when I have to make the gametes, from this pair, either capital R can come and from this pair, capital Y can come. This is one type. Next type, from this pair, small r can come. Okay, from this pair, capital Y can come. Next thing, from this pair, capital R can come. From this pair, small y can come. Okay, similarly, from this pair, small r can come. From this pair, small y can come. So these are the type of gametes that I get from this parent. 4 type of gametes I get from this parent. Similarly, 4 of them I will be getting from this parent also, which would also be similar. This. So just make contrasting. From here, choose one, here, choose one and make. Okay, so first do the dominant ones if it is easy for you. Okay, either way you make the gametes over here. Now you have to fuse them, so you cannot cross it like this and show. Instead you use something that is known as Punnett square, where you put the gametes on one side, the male gametes, the female gametes. Okay, and then you cross. So if you see over here, if I zoom it in, I can find something like this. If I cross this with this, I get capital R, capital Y, capital Y. If I fuse with this, capital R, small r, capital Y. If I fuse this with this, I get capital R, capital Y, small y. In this way, I have to cross them, children. So this is known as Punnett square. What do I call it? It is known as Punnett square. Now, one very important thing. In F2 generation, they will ask you the phenotypic ratio. They will not ask you the genotypic ratio. They won't ask you because there are so many genotypes that can be formed over here and will be really difficult for you to remember it. So they will ask you something that is known as phenotypic ratio. What is phenotypic ratio? 9:3:3:1. So you will be finding 9 plants which are round yellow. Okay. 3 of them which are round green, phenotypic as in how do they look. 3 of them which is wrinkled yellow and 1 of them which is wrinkled green. And how will you remember it? It's a very easy thing, children. Just try to understand what I am trying to say over here. 9:3:3:1. 9 goes for the parental dominant, that is round yellow. Okay. 1 goes to the parental recessive one, which is wrinkled green, which we started off as a parent. Now you have to make new combination. So round one is with yellow over here. Let's make it with green. This is what this 3 stands for. Now this 3 stands for the wrinkled one. So wrinkled was with green. Let's make it yellow. So these are the new combination that I can find. This is the new combination that I can find. 3 will be round and green and 3 will be wrinkled and yellow. So I am able to find newer combinations also in F2 generation. Okay, such that which was not found in the parental generation. And what does this show? This shows the next law, which is known as law of independent assortment. According to it, what is it? The alleles for one gene sort into gametes independently of alleles of another gene. That is, when you talk about okay, these are alleles of one gene. These are alleles of another gene. I am saying they are going to independently assort into the gametes. So whenever gametes are formed, either this chromosome can go or this chromosome can go. If this is going, so suppose R is going. Over here, this chromosome can go along with it or it can go with this. So it can be either like this or it can be either like this also. You see, they are not saying that capital R will go with capital Y only. It is not the case. So I will see that independently assorting. They are independently gain segregated over here. If independent assortment was not there, then every time round one will be with yellow only. every time round 1 will go with yellow. So you won't find new variations. You are finding variations because this is happening. They are independently sorting. This is going with this is going with this. Also similarly, this goes with this goes with this also. Once more we will read it. It says that alleles for one gene, okay, sort into gametes independent of the alleles for the another gene. This is known as law of independent assortment, and every time question comes, that is, they give you the scenario and they will ask you what law is being shown over here. This is the law of independent assortment. So in dihybrid cross, you always associate with law of independent assortment. Works. So these were the 3 laws given by Mendel. With this, we are completing the crosses also. Now comes sex determination in the case of human beings. Okay, how does this happen in the case of human beings? So we say that we have 23 pair of chromosome, my dear children. How many? We have 23 pair of chromosome. Among that, 22 of them, they are known as autosomes. They are known as autosomes. So 22 pair are known as autosome. 23rd pair, they are known as sex chromosomes, or what I call them allosomes. Sex chromosomes or allosomes. Okay, so sex chromosome, basically it can be either X and X as in the case of females, or it can be X and Y as in the case of males. So either if this is the case, then it will be female. If this is the case, that it will be male. One thing that you should be knowing is Y chromosome is shorter than X chromosome. So if they are saying that there is a shorter chromosome along with X chromosome, just understand you are talking about Y chromosome and that person will be a male. Okay, so mother will be having XX, the father is having XY. So the gametes that will be formed over here, whatever you do, it will be X only in the gametes. But here you see the father, there are 2 types of gametes being formed. Either it can be X or either it can be Y. Now if X and X comes together, it will be XX. If X and Y comes together, it will be XY. If X and X comes together, it will be XX. X and Y comes together, it will be XY. So how much % chance is there for a boy child to come and for a girl child to come? It's 50-50 chances. That is, this 50% chance. This is also 50% chance. Got it? Works? Okay, so this is a question that has been asked. Is it correct to say that women are responsible for the sex of the child? No, it is the men who are responsible for the sex of the child, even though I cannot say it's truly correct, but scientifically if you are saying yes, none of them should be responsible for the sex of the child and should not be a big deal also. But okay, scientifically if you see, there are 2 gametes being given by the men, which is why this is a question, okay, and this comes usually every single time. So you should be saying that according to scientific basis, the father will be having 2 types of gametes, which is why it should not be put on the mother. The blame should not be put on the mother or the females. You have to explain it like, make the cross like this, put parents, gametes and then F1 generation. I know it's totally wrong to blame anyone, but yes, this is what our NCERT says, just to avoid, because so many people were blaming females for this, which is unduly wrong. So we should not be doing that. We should not be blaming any gender, and the sex of the child should not be a big thing for any of us, because we are educated and we believe in equalism, right? So equally, both of them are really good. So yes, but scientifically saying, this is the thing. Cool. Now sex determination is different in the case of other reptiles. Okay, there are certain reptiles which do sex determination with the help of temperature. For example, they lay their eggs. They lay their eggs and put it somewhere where the temperature is really less. If the temperature is really less, that is mostly below 31 degrees Celsius, it comes out to be a female. If there is high temperature, it will come out to a male. So this is known as temperature dependent sex determination. This happens in certain reptiles, for example, crocodiles, certain type of lizards and turtles and stuff like that. So sex determination is different in different type of organisms. One such example is given as this, okay, in your textbook about reptiles, so please understand it. This is known as temperature dependent sex determination. The other one is about snail. Snails, basically they can change their sex. For example, males can change into females as well. Okay, so that is another type of sex determination. So we have XX, XY type of sex determination. Other organisms have other types. Please be remembering that. Now one last thing that is given in your NCERT textbook is about blood group. There is no explanation regarding that, but they have given the questions, so we should be knowing it. So according to blood groups, so until now we said that height is regulated by 2 alleles, that is capital T, small t. Over here blood groups are regulated by 3 type of alleles. One is IO, okay, that stands for O blood group. One is IA, that stands for A blood group. One is IB, which stands for B blood group. Okay, now suppose I say that a person has A blood group. What all can be the genotype? What all can be the genotype? It can be IA, IA. So in one chromosome it can be IA, other chromosome also it can be IA. Okay. Next scenario can be, it can be IA and IO. This is also genotype. That means A is dominant over O, which is why A is being expressed over here. Understand? So A, I am writing A is dominant. A is a dominant allele. Okay. Now for a person to have B blood group, it should be IB homozygous, or it can be IB IO. That means B is also dominant over O. So I will say B is also dominant. Now for person to have O blood group, it should be IO. Either of these guys, if they come, it will dominate. So this is a recessive allele. And now you might be having this doubt, ma'am, there is AB blood group also. So how do you get AB blood group, children? When a parent gives A, another parent gives B, over here you'll get AB. So ma'am, isn't it dominant? No, they are kind of co-dominant. What do you call it? Co-dominant. Works? Okay, so how the questions come, children, they will be giving you, probably they will saying the mother has A blood group, what is the and the father is B blood group, what will be the genotype, or basically what is the blood group of the child. If mother is A blood group, children, if the mother is A blood group and the father is B blood group, the mother's genotype can be either IA, IO or IA, IA. Okay, and the father is B blood group, that means again it can be IB, IO, or it can be IB, IB. So you have to probably make for all. So you have to check, and that is a humongous task, but you have to do it. What are the possible blood groups that child can have? Basically you have to cross this with this. So you will say AB is there, okay, IA, IB, then you have cross with this. So you have to cross this with this, you have to cross this with this, you have to cross this with this, you have to cross this with this. Either way, at the end, whatever possible blood groups are possible, you have to tell. So suppose when I cross both of them, what do I get? I'm just making an example. I've explained in detail, so go watch the video there. So this crosses with this, I get A. Okay, when I cross this with this, I get IB, IO. When I cross this with this, I get IO, IO. So in one cross only I was able to find that there is AB blood group possible, there is A blood group possible, there is B blood group possible, there is O blood group possible. So in this I can say directly that these many blood groups are possible, and whatever you do anyway, they will be included. One of them will be included. So these are the possible blood groups that the child can have. Okay, the possible blood groups that the child can have. So if you have, you will be using it. Okay, so do that, and if they are asking for a big explanation, you have to cross each and everything and show. Okay, if they are not asking for a big explanation, it's a 2-marker question or something like that, then you just have to make a cross and just understand that this is the case. Okay, but please write parent, then form gametes also. Here I didn't make the gametes, these will be the gametes, this will be the parent. So make like that. Okay, don't go skip the steps. Works. Now one more thing is there about the earlobes, which is given in your NCERT. Please understand, free earlobe, okay, the one that is not attached, is a dominant character, is a dominant character, free earlobes, whereas the attached one, just like how I am having, it is recessive character. Okay, so suppose F stands for dominant, this one, and this stands for recessive. So probably my father and mother have something like this, which is why both of them have free earlobe, both of them have F2 dominant. But when I was born, these are the gametes that could be formed, right? Probably these two gametes came together, which is why I have the recessive ones. So it's not like two parents with free earlobe cannot have a attached earlobe child. They can have it. Their condition could be like this. Their condition could be like this, which is why they can show up the character. So this is all, my dear children, for revision for biology. I tried to make it as fast as possible, but I know I could probably have failed, but I tried to include everything that was there. Just remember, there is one more thing that is known as test cross. Test cross is basically when you are not aware about, for example, you got it can be this or this. This is the generation. Now you are not aware. You want to find whether it is homozygous or heterozygous. What do? You cross this with the recessive parent, that is small t. This is known as test cross. Okay, this is known as test cross. If you want to know more about test cross, you can just put it down in the comment section. I could probably make you shorts for it, but you have to tell me. Yeah, and now if you are someone who is not aware about what you should be doing after 10 children, you have Diksha's Scholastic Aptitude Test, which you can take. Okay, the link to it is given in the description box. It will be a 60-minute concept based lecture, after which there will be an aptitude test based on the lecture. Okay, and then a one-on-one counseling, where the counselor will suggest to you the best stream that will be good for you. So that is all about it. So if you're someone who is interested, you can click it there, and please wait for the revision by physics sir. He will be continuing this particular class. So that's all. Bye-bye.
So hello students, Jasmine ma'am has completed her part in the biology section. Now it is my time to revise the content of the physics. Okay, so what we will do, first we will revise the chapter quickly, what are all there, then we will revise the concepts quickly, and that's it. Tomorrow get ready for the test. Okay, so first of all, as I said, the chapter revision. So first the human eye, okay, power of accommodation, then we have defects of vision. How many defects of vision there are? There are 3 different types of defects of vision: myopia or nearsightedness, hypermetropia or farsightedness, and then we have presbyopia, okay, that occurs due to age. Then we have natural phenomena. In the natural phenomena we have dispersion of white light, then we have atmospheric refraction. In atmospheric refraction we have twinkling of stars, twinkle twinkle little stars, right, advanced sunrise and delayed sunset. Then we have scattering of light, Tyndall effect, and blue sky, why the sky appears blue. So everything will be covered quickly. Whole chapter we will cover quickly. Then tomorrow will be your test, and Monday I will do the PYQs from this chapter. Okay, great. Now let's start with the accommodation of eye part. So the ability of the eye lens to adjust its focal length is called accommodation. So as we know, our eye lens can adjust the focal length so that we can see distant object as well as close by object using the single lens, which is not possible using a single lens for a physical lens, right? But our eye is amazing. You have to take care of your eye, because it can help you to see distant object as well as close by objects, right? And that phenomena is called accommodation. However, the focal length of the eye lens cannot be decreased below a certain minimum limit. That is called minimum distance of distinct vision. What is that? For a young adult, the normal vision, the near point is about 25 centimeter. Okay, 25 centimeter is the distance up to which we can see. Right, this is the minimum distance. Minimum distance of distinct vision is 25 centimeter, and the far point is infinity. Far point is obviously infinity. We can see up to infinity with a normal eye. Thus, a normal human can see an object between 20 centimeter up to infinity. Okay, from 25 centimeter up to infinity, a normal human being can see. Okay, like you, like me, I need a glasses, but still I can see from 25 centimeter up to infinity after wearing my glasses, right? Now next is the common refractive defects of vision are myopia or nearsightedness, hypermetropia or farsightedness, presbyopia. Okay, myopia or nearsightedness means a person can see nearby objects clearly but cannot see distant objects clearly, so that is myopia. So let's see, a person with myopia can see nearby objects clearly but cannot see distant objects distinctly. Okay, causes, what are the possible causes? This is the summary of the entire thing, and important for exam, so don't miss it. Excessive curvature of the eye lens. If the eye lens is excessive curved, okay, then maybe you have myopia. Elongation of the eyeball can be elongated, and thus the rays are forming even before they are reaching your eye. That's why you need a concave lens to correct your vision. Okay, your eye power has increased. Some say I have a power in my eye. Okay, or some say my power is increased. Actually, figuratively, it's true if you have myopia. Basically, what happened is that your eye lens is now more powerful, and because of that, the rays are actually forming even before they are reaching the retina. So you have to decrease that eye power by using concave lens. Okay, concave lens is something which diverges rays. We know that. So because of this small divergence, okay, your eye power is reduced and your eye power becomes normal. right? Now next is what? Next is hypermetropia or farsightedness. What is that? A person with hypermetropia can see distant objects clearly but cannot see nearby objects distinctly. Okay, so they can see distant objects which is very far clearly but cannot see close by objects. So while reading books or watching movies or playing video games, they face difficulty. Okay, so someone is playing PUBG, instead of shooting the enemy, he shoot the friend because he has nearsightedness. So that is a very farsightedness. That is a very unfortunate thing. So what are the reasons? Reason is the focal length of the eye lens is too long. Okay, the focal length is increased, right? So that is one reason. And the eyeball has become too small. So eyeball size of the eyeball is too small. That's why the eye is not able to see close by objects. This defect can be corrected by using a convex lens of appropriate power. Okay, now what you have to do, because the eye light rays from close objects are focusing beyond your retina, now you have to use a convex lens to adjust this and make sure that rays are forming into your retina. Okay, so this is the basic thing about hypermetropia. Last thing is presbyopia. Presbyopia generally occurs with age. The power of accommodation of the eye usually decreases with aging because your ciliary muscles lose the traction. They are not flexible enough. Okay, and that's why eye cannot accommodate distant objects and close by objects, and this is caused due to age. The farsightedness and nearsightedness both can occur simultaneously. So in case of presbyopia, person can neither see close by objects nor see distant objects. Okay, somewhere in between he can see slightly, otherwise he or she cannot see. So that is a very difficult problem to deal with, and the remedy for this to use bifocal lens. Okay, remedy is use of bifocal lens. A bifocal lens, as I said, they have two distinct lenses. One is convex lens for reading close by things, and there is a part which helps you to see distant objects. Okay, both type of lenses are available, and that is a bifocal lens. Next is homework problem. Let's solve the homework problem I gave you. What was the homework problem? Study the diagram below and answer the question that follows. Name the defect of vision depicted in the diagram, stating the part of the eye responsible for this condition. So basically, as you can see, the nearby objects, the near point is shifted far. Okay, and the objects he cannot see objects which is closer to his eye. So basically it's a hypermetropia. Okay, the defect is hypermetropia. And possible reason, what are parts are responsible? Basically the lens, eye lens. Two parts can be responsible. Eye lens is elongated, right, or eyeball is too small. It is not clear from the diagram, so you can write both of the reasons. Okay, least two causes for the defect. These are actually causes for the defect. Okay, you can mention in the first part, you can mention eye lens. Okay, only eye lens is sufficient. Next is name the type of lens used to correct the defect, state in the role in this case. So we need a convex lens. Okay, we need a convex lens to make sure that the rays coming from this are actually focused on the mirror, and they appear come from here. Okay, so basically what is happening, sorry, my mistake, this is not the ray diagram. If you use a convex lens, what happens is ray from here so they appears to come from here. Okay, so you need a convex lens to correct this kind of refractive error. Why? Because your eye is no longer able to focus them on the retina. So the thing is that convex lens is making the rays more convergent so that the ray can meet at the retina. That's it. Okay, so this is the role of the lens. Okay, I hope this helps. Next question: a student sitting at the back of the classroom cannot read clearly the letters written on the blackboard. What advice will a doctor give to her? Draw a ray diagram for the correction of this defect. So ray diagram I have already shown you. Okay, the first part of the question is, it is a myopia, and the myopic eye, for myopic eye, what is the ray diagram? I have shown you. You have to draw that ray diagram, and you have to mention that use convex lens. The doctor will advise her to wear glasses of concave lens of appropriate power so that she can see the objects clearly which is behind. Okay, and draw the ray diagram. You have to draw the ray diagram using the concave lens. Okay. Next part is, if you are also not able to see your future, which is very far, and you are thinking I will just sit back, relax, play PUBG, don't do that, because your future is very, very important. And to know what is the future for you, you can appear for Diksha Scholastic Aptitude Test, where we will be giving you 3-step process to clear your future goals. Okay, to clear in your mind that what should be your future goal. What are all the 3 steps? First step is a lecture. Okay, there will be 60-minute concept based lecture. After the lecture is over, there will be doubt clearing session. Once that all the doubts regarding you is cleared, then what you can do, there will be an aptitude test based on that class. Okay, based on that lecture. Once the aptitude test is over, based on your result, there will be a counselor. He or she will guide you exactly what your future looks like. Okay, and they will also guide you what you should focus on in future. You are good for joint entrance to take admission in IITs, or you are good for NEET to take admission in any of the AIIMS, right? Or you should prepare for KSET to take admission in one of the Karnataka's leading colleges, right, for engineering. But for that, you have to fill up this form, and our representative will contact you with further details about this Diksha Scholastic Aptitude Test. Okay, now if you are satisfied, let's move on to the next part, where we are going to discuss about the splitting of white light into 7 colors, that is dispersion of white light. So what is dispersion? The splitting of light into component color is called dispersion. So as the white light passing through a prism, after it emerging out, it splits into 7 constituent colors. What are all the 7 colors? Violet, indigo, blue, green, yellow, orange, red, from bottom to top. Okay, from base to top. Okay, this is the base of the prism. I already told you this is called base. Okay, and if you see VIB Gior, okay, so this is the thing that you should remember. Okay, VIB, Viv, G is for green, Gior, Y is for yellow, O is for orange, and R is for red. Okay, clear? Different colors of light bends through different angles with respect to the incident ray. So when the light is falling on this prism, different ray of light bends differently, because they all have different refractive index, because the refractive index depends on the light of the color, right, frequency of the light. So that's why different light bends different angle. So if you remember, during the definition of refractive index, we said monochromatic light. Monochromatic light means only one color of light. So when we say the refractive index of a glass is 1.5, that means that represents a particular color, generally yellow. Okay, most of the cases where you don't see any light, that means it is basically given for yellow light. Okay, they will not mention it, but mostly it is for yellow light. But if you are using it for red light or violet light, the refractive index will vary. Okay, respect to the incident light, as they pass through prism, the red light bends the least, whereas the violet light bends the most. Okay, so violet light bends most, red light bends least. Okay, and that's why you get red at the top and violet at the bottom. Next is rainbow. So what are the conditions to observe rainbow? We have learned everything about rainbow yesterday. So there should be a water vapor present in the air. Okay, so air to observe rainbow, two necessary conditions are, first of all, there should be water vapor present in the air. What is the second point? Second point is the observer should look opposite to the sun. So observer, if you want to observe rainbow, sun should be in your behind and you should look opposite side of the sun to observe the rainbow. You cannot see the rainbow in the same side as the sun. Clear? These 2 points are very important for your board exam. Next is atmospheric refraction. So twinkling of stars, we have seen twinkling of stars. Atmosphere has constituting of multiple layers, and multiple layers have different refractive index. So when ray of light traveling from outside earth towards earth, okay, what happens is that the light rays fluctuate rapidly, and because the stars are very small and small amount of light is coming from the stars, once they reach our eye, we do not see their actual position, but we see them slightly upward direction, right? Now because of that, what happens is that this apparent position changes with time, okay, because of the fluctuation in the atmospheric refractive index, okay, and thus the stars appears to be twinkling. But the planets which are much closer to the earth, they can be you can consider them to be object or having multiple extended source. Okay, multiple sources. Extended source means they have light coming from more than one point. Okay, there is a point source, there is an extended source. Point source means like the tip of an incense stick. Okay, if you have seen an incense stick, the top of the incense stick, you can consider it to be a point source or a laser pointer. Okay, so very small laser pointer, they can also be considered to be a point source. What is an extended source? Normal tube light, normal LED light, normal torch light, they are all extended sources. Okay, the planet is like an extended source. So when even after atmospheric refraction, their positions average out and you do not see any twinkling of the planets. Okay, so if you in the night sky, if you see two stars appearing in the sky, one of the star is twinkling, another star is not twinkling, that means the star which is not twinkling is not probably a star, that is a planet close to earth. Okay, and the star which is twinkling, that is a star far away from us. Okay, so that's how you can distinguish between stars and planets. Okay, next is atmospheric refraction. The sun is visible to us about 2 minutes before actual sunrise and about 2 minutes after the actual sunset, because of the atmospheric refraction. Even if the sun has gone down the horizon, okay, we can see the sun in the sky because of the atmospheric refraction. Okay, if you are sitting in a mountain place, you see that the sun is going down. Actually, sun has already gone down. Okay, if the sun is just touching the horizon, maybe in a seashore, okay, you are sitting in a beach and you are observing the sunset, then probably the sun that you are seeing to be setting, actually it has already happened. Okay, and that's why when the sunset happens, immediately there is a complete darkness because sun has gone down a lot and we were just seeing the reminiscent of the sun. Okay, this is a very interesting thing, and you should remember that you are getting 4 minutes of extra daylight every day because of this, right? Now what next? Scattering of light. Tyndall effect, we have learned about there are two different types of effect. One is Tyndall effect. So if you light up a medium having colloids inside it, okay, the colloids reflect the light and they appear brighter, that is called Tyndall effect. Okay, the phenomena of scattering of light by the colloidal particles gives rise to Tyndall effect. It is a very beautiful effect, you can try it at home also. When the sunlight passes through atmosphere, the fine particles in air scatters the blue color, okay, and more strongly than red. So that's why we see the sky blue. Okay, so as I explained, suppose there is a cloud, okay, sunlight is falling on the cloud. After scattering, red is almost go undeflected, but blue color scatters the most, and you are standing here, so blue light is reaching to your eye, whereas red light passes straight almost without much scattering. Okay, that's why you see the sky to be blue. That's why sky appears to be blue, and that blue color is reflected by ocean. That's why ocean appears blue as well. Okay, so the color of the ocean, color of the sky, both are related to the atmosphere is scattering. Okay, even though ocean is not directly related, it is because the ocean reflects sky. Okay, that's why it appears blue, whereas sky appears blue because it scatters the blue light. Okay, so understood. I hope you have enjoyed this class, and you will enjoy our DSAT orientation, where we are taking a Diksha Scholastic Aptitude Test that is basically consisting of 3 parts. As I mentioned, first part is a 60 minutes concept based lecture video, concept based lecture, one-to-one lecture. It is a classroom based lecture, not one-to-one, classroom based lecture. Once the lecture is over, any doubts that you might have, the teacher will clear all the doubts that you might have. Also, after the lecture is over, there will be an aptitude test. Based on that test results, there will be an one-on-one counseling for your future. Okay, what is good for you, the counselor will tell you what is good for you, why that is good for you. Okay, and whether you should opt for any of this in future, whether you should try to get a rank in JEE, or you should more focus on NEET, or you should focus on KSET. Okay, so based on this, there will be a very interesting career path for you, tailor made only for you. And only thing that you have to do, you have to fill up this form. The link of this form is given in the description. Don't miss out this opportunity. It is happening across our Diksha campuses every Sunday. Okay, and if you have liked our video, including Jasmine ma'am's and mine, then don't forget to like, share and subscribe this video with your friends.